Today we're going to present about the Hedgehog signaling pathway. My group members are Ng Tian Yu, Ho Chi Siang, Dwayne, which is me, and Ted Mak Tu. What is cell signaling? Cell signaling is a process where cells interact with one another and react to outside stimuli. It also controls a variety of biological processes such as immunological response, growth, development, and homeostasis. There's four examples of cell signaling, which are autocrine, paracrine, endocrine, and juxtacrine signaling. Moving on to hedgehog signaling. The purpose of hedgehog signaling is for embryonic development. It plays a crucial role in the proper formation of tissues during the development of the embryo, causing and influence cell differentiation and organ development. It also for tissue repair, neural development, and cell growth. If there's any abnormality in the cell signaling, it will cause development disorders such as syndactyl, which is the binding of the fingers being fused together, and also skin cancer.
So now I'll be talking about the reaction mechanism of the hedgehog signaling. So hedgehog signaling has two states, the off state and the on state. In the off state, HH is not bound to patch 1. HH is the signaling molecule and patch 1 is the receptor for HH, but it's not here. So because it's not here, this inhibits SMO translocation into the primary cilium. SMO is smoothened and it's a signal transducer. This inhibition induces phosphorylation of sulfur and Gli. Sulfur stands for suppressor of fused and is a negative regulator of hedgehog signaling, while Gli mediates transcriptions of genes in the entire pathway. So this phosphorylation is caused by PKA, CK1, and GSK3B. PKA is protein kinase A, CK1 is casein kinase 1, and GSK3B is glycogen synthase kinase 3 beta. So the proteins are further degraded by BTRCP or beta transducin repeat containing protein to form repressor forms, but because they're repressed when they enter the nucleus, there's no transcriptions of the target genes. So in the on state, HH, which is hedgehog, is actually bound to patch 1 over here. So this binding releases smoothen. So smoothen can enter the primary cilia, and this causes beta-arrestin to accumulate, thus activating the Gli transcription factors. So the transcription factors then get activated, they get cleaved into their activated form, and then they go into the nucleus to transcribe the genes. So target factors like Gly1 and patch1 are produced so the negative and positive feedback loop can continue, while MYC, cyclin D, and cyclin E over here are created for cell proliferation, while BCL2 helps with cell survival.
In my slides, I will be talking about Gorlin syndrome. Gorlin syndrome is a rare type of basal cell carcinoma caused by the mutation of the patch1 gene. Under normal circumstances, the patch1 gene is a repressor of the hedgehog signaling pathway. When hedgehog ligands are not present, they prevent the activation of the smoothened protein, preventing the continued proliferation and differentiation of cells, especially overactivation, which causes the growth of tumors. When the patch1 gene doesn't function, it allows the hedgehog signaling pathway to overwork and eventually cause the growth of tumors. It affects 1 in 40 to 60,000 babies equally across both males and females, and 70% of cases are from inheritance, with 30% being more spontaneous mutations. Now, homozygous inactivation of the patch1 does not necessarily lead to immediate cancer, but it does show symptoms within early life, where we're talking about skeletal abnormalities such as macrocephaly, as seen here, polydactyly, extra fingers, rib abnormalities, and facial malformations. Typically, those who have a single mutated patch1 gene will require a second mutated gene from cases such as UV light and ionizing radiation for the growth of tumors to start beginning. But throughout their entire lives, any patients with Gorlin syndrome will always have increased risk of other cancers such as medulloblastoma and melanoma, which is a type of skin cancer.
As said earlier, Gorlin syndrome is caused by a mutation in the patch1 gene. A potential solution is a new modern technology called CRISPR-Cas9. CRISPR-Cas9 is a relatively new technology used to edit parts of the genome. This can include adding, removing, or altering specific sections of the DNA sequence. So how does it work? First, a guide RNA can be designed to target the mutated region of the patch1 gene. Guided by the guide RNA, the Cas9 protein then creates a double-strand break in the patch1 gene. The patch1 gene can be repaired via non-homologous end joining or homology-directed repair. So, non-homologous end joining fixes the mutated gene by creating insertions or deletions which disrupts the function of the mutated gene. However, homology-directed repair utilizes homologous regions as templates to fix the mutated gene error-free. So, the mutated patch1 gene can be completely fixed and reversed to its original function using HDR.